My lab is interested in attachment. How do we encode the social memories that define relationships throughout our lives, and how are they disrupted in the context of psychiatric illness? So, social attachments manifest in every aspect of human life, whether we're talking about the attachment between parents and children, between family and friends, and subsequently conflict or competition between groups or individuals, and that most notorious form of social attachment, romantic love. Now, equally pervasive, not surprisingly, is the disruption of social attachments across the spectrum of psychiatric illness, whether we're talking about the isolation of depression, the incredible grief that can be associated with, for example, traumatic loss, the confusion and the disruption of social attachment and social skills in the context of psychotic illness, or in the context of personality disorders. And now, not surprisingly, for this audience, the unifying principle between these healthy and pathological disruptions in social attachment is that they're a consequence of not only a human brain, but human brains interacting with each other. And so then the question is, how can we understand how relationships are encoded and how they then translate into future behavior later in life? Now, in addition to just simply understanding the neurobiology of the encoding of social attachment, as a child psychiatrist and a developmental neuroscientist, social attachments also offer a really powerful system to understand how early experience can very dramatically impact life at later stages of development, in particular in adulthood. And this allows us to watch the development of circuits throughout the life of an animal to really understand, for example, how early traumatic experiences can disrupt adult patterns of attachment behaviors. Now, humans fall into the category that we call socially monogamous. That's about 4 to 5% of mammals. They exhibit pair bonds, or strong relationships between mates, and this is thought to facilitate biparental care. Mates will actually seek proximity to each other. They will show a preference for their partners over other individuals, and this is generally thought to facilitate reproduction. At a larger level, this leads to the formation of family units, again with biparental care, that are thought to provide nutrition, protection, and then through the cultural transmission of knowledge, information about social interactions at large. An important feature of social attachments is that when separation occurs, there's an anxiety-like response. Now, as a neuroscientist, it's important for me to communicate to you that mice, which are the predominant model system for mammalian neurobiology, don't display adult attachment behaviors. They don't display pair bonds, and generally don't display biparental care. And as Brandon introduced, hence the prairie vole. So prairie voles are small rodents, slightly bigger than a mouse, that again display social monogamy. Both in the wild as well as in the laboratory, mates form pair bonds, seek proximity to each other, show a preference, and an anxiety response to separation. This is thought to facilitate both parents defending the nest, providing parental care, and then at a larger social level, prairie voles also display alloparental care, or the care of offspring that aren't their own. And a very interesting evolutionary correlate of monogamy is incest avoidance. So just to show you how robust these behaviors are, and because I can, I'm just going to show you a very quick video of what prairie voles look like in the laboratory. So what we did is we took 2 animals that had been bonded for a couple of months, removed, in this case, the female, you can do it with either the male or the female, for just an hour, and then introduced her back into the cage. Following that introduction, what you see here is that the female is wandering around the cage, the male in the back very politely escorts her back to the nest, and the 2 will engage in this incredibly robust huddling behavior, basically side by side, as much contact as they can get for the duration of the assay. And I'm not going to talk about the details, but Shuyu Wang, another MD PhD postdoc in my lab and psychiatrist, as well as Kara Quine, a computer scientist from Microsoft, have developed machine learning and video tracking approaches to be able to analyze quantitatively and track these animals' behaviors without the bias of human observation. Now, pioneering work from Sue Carter, Lowell Getz, Tom Insel, Larry Young, and others demonstrated that the potential for social monogamy correlates with the ability of 2 hormones to work in different parts of the brain. Those hormones are oxytocin and vasopressin. And the take-home point here is just if you look at this monogamous prairie vole versus this promiscuous, in this case a montane vole, you see that they're very, very different patterns of binding of oxytocin. We don't need to get into the details, but suffice it to say, this really implicated oxytocin and vasopressin as some of the key regulators of the formation of social attachment. And an interesting feature of prairie voles themselves is that within the species, you see that there are some animals that have high and some animals that have low levels of oxytocin binding in this case, and that correlates with their fidelity when we actually test them in the laboratory setting. Again, just in support of the role of oxytocin and vasopressin, if we take animals and just let them hang out for a short period of time, they don't really show a lot of interest in each other. They're a little bit antagonistic, not terribly friendly. But now in the same period of time, if we give them either vasopressin or oxytocin, we can induce this huddling behavior. So again, that really speaks to the important role that these hormones play in the mediation of social attachment and pair bond formation. Now, the important fact that allows us to really sort of translate this work into clinical settings is that the role of oxytocin and vasopressin is really conserved across phylogeny in terms of their mediation of different aspects of social behaviors. That doesn't mean that all animals show attachment, but in many cases, we find that oxytocin or its related hormones modulates different aspects of social behavior in very, very diverse species. So what this all led to is the hypothesis that oxytocin and vasopressin are key nodes to really understand how we form relationships and what parts of the brain are involved in those. But when we started this work, we really didn't have the ability to manipulate the prairie vole brain in the same way that we're used to thinking about all of the techniques that we use in mice to really understand the circuits and the molecular biology underlying social attachment, and then thinking about how they're disrupted in the context of psychiatric illness. And so two postdocs in my lab really pioneered the work using CRISPR mutagenesis. I'm not going to get into the details, but suffice it to say, it's basically a molecular laser that allows you to manipulate the genome of almost any animal. And so Kristen Berenson, another MD PhD, and Ruchira Sharma used this approach to understand the role of oxytocin receptor signaling by generating animals that lack it. So now, based in this system, what is our question? Our question is to really understand what are the sensory and neural mechanisms that mediate social attachment. Why is it that if you were to look around the room and make eye contact with a stranger versus with a friend or a loved one, it evokes a very, very different response within you? How do we go from first sight to whatever happens next? So now when we think of the brain, not surprisingly, there are many, many, many different pathways, from our eyes themselves, we're talking about humans, into the brain in terms of understanding and processing the sensory information, and then driving behavior. And you can imagine that psychiatric illness can result from disruptions in any part of this circuitry. And so and what we also know is that patients, for example, with schizophrenia or with autism, have different patterns of tracking social information. So for example, where in the face they pay attention to, or the tracking of what's called biological motion. And so this again suggests that in the context of psychiatric illnesses, there are disruptions to the processing of socially relevant sensory information. So I'm just going to quickly go through a set of experiments that one of my grad students did. Narissa Hoagland used a setup where we test the preference of an animal for its partner versus stranger, and find robust preference for the partners in bonded voles. But then what Narissa did was to actually separate the animals using sort of jail cells, where they can't necessarily touch each other, but they can see, smell, and hear. And she found in that context that males can no longer differentiate partner versus stranger, whereas females can. And then because Narissa is very interested in auditory processing, she put earplugs into the females in the same context, and found that now the females can no longer identify partner versus stranger, which means that obviously females can use auditory information, but more importantly, that males versus females use different types of sensory information to identify their partners. So now turning to the animal model, the other thing that we want to understand is not only what are the sensory cues, but how does the brain process this information? And because we're dealing with rodents, our first hypothesis is that smell or olfactory cues are an important part of how animals differentiate with each other. Not getting into too much of the details, what we focused on is a region of the brain called the medial amygdala that processes pheromones, right, which are very important for animal behavior. We used a system called fiber photometry, basically using the ability to look at bulk calcium activity, which reflects the activity of neurons in this region of the brain, to look at what happens when animals interact with partners versus strangers. And the important thing to take home from this is that we see very, very different patterns of response when an animal sniffs its partner versus a stranger. So shown in black, indicated by P, we see that when an animal is sniffing its partner, there's a really high level of activity before it sniffs, followed by a suppression of activity in the medial amygdala after, versus for a stranger, there's not a lot ahead of time, and then suddenly there's a lot of activity after the animal actually sniffs a stranger. And so using the Inscopics portion of this grant, what we can now do is go into the medial amygdala and look with single-cell resolution at how these sensory cues are actually being encoded. Are there changes to the partner-responsive neurons, the stranger-responsive neurons? What happens to the neurons that respond to both? How do they change over the course of pair bonding? And we can use this system to, again, this is just a recording in the medial amygdala of neurons firing during behavior, and then you can see a trace of individual cells correlating with the vertical lines, which are the individual behaviors that the animal are actually engaging in. So finally, the big question is, how is the processing or detection of these sensory cues disrupted in the context of psychiatric illness? And so again, we turn to the CRISPR-Cas9 mutagenesis that we developed in prairie voles, and for the purposes of this project, are looking at mutants in two genes. SHANK3, which is a gene that's been genetically associated with both autism and schizophrenia in human populations, as well as the dopamine D2 receptor, which has both been associated with schizophrenia, but also is the principal target of most antipsychotic medications. And so you can imagine there's an important question as to what are the effects of these drugs in the first place on baseline behaviors, and then how does that actually translate to changes in social and attachment behaviors. So just to show you one little tiny piece of data, a grad student in my lab, Rose Larios, looked at the SHANK3 mutants and found that females lacking one copy of SHANK3 show deficits in social attachment behaviors. And this just becomes really relevant because, first of all, again, we see a difference between males and females. And more importantly, these females are only lacking one copy, which is like what we see in human populations. They're not lacking every bit of SHANK3. So just to end, what I hope I've convinced you of is that sensory processing of social cues is a really important part of how we understand our ability to form and maintain social relationships. These are disrupted in the context of various psychiatric illnesses, and we now have a model system to really tease out what the underlying neurobiology of attachment behavior and these sensory cues is. Just want to end by thanking my lab, our collaborators, and our funding sources. Thank you very much.